हेलो ठीक हेलो
हेलो कोड दिया है हेलो हेलो रह এইটা এটা তো এইটা দিয়ে আসছি हेलो 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 हेलो
हेलो
हेलो
এইটা কোন তো আইনি তো
কোডা हेलो এইটা না সেইটা এগুলো ঠিক हेलो चेक
ओके साउंड
चेक ତମ ସାଉଣ୍ଡ ଆସୁଛି ମୋ ସାଉଣ୍ଡ ଆସୁଛି କି ନା ସେଇଟା ନାହିଁ ତମ ସାଉଣ୍ଡ ଆସୁଛି ମୋ ସାଉଣ୍ଡ ଆସିବା ଦରକାର ନା हेलो हेलो चेक हेलो हेलो हेलो
हेलो सर सर ಏನು ಮಾತಾಡಿದ್ ಮಾಡಿದ್ರಿ हेलो
yeah hello
sir am I audible to you
yeah you're audible
okay sir thank you
हेलो चेक चेक हेलो चेक चेक हेलो चेक हेलो चेक
কোডা লাগিছে কি
Check.
আছে কি এ তারটা কোড লাগিছে দেখিলু এসে লাগাছে এইটা এটা তো
हेलो हेलो चेक हेलो हेलो चेक हेलो 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 ना हेलो I'm audible check हेलो
চলো
हेलो
चेक चेक
we should coordinate because I don't know how. Yeah, 50 minutes for each speaker. And she is told that the speaker will have Leonardo's full formation and that she's not here. And Chitra Kulkarni will be joining online. And other person also online. A speaker. So I think
भाई এটা কাম করবে ভলিউম আসুন Yes, better. Slides play करना
Hello. So we're going to have this parallel session of day one. So this time constraint, the Honorable Chair has decided that the session will be 8 plus 2 minutes. So all the participants
are requested to deliver their lecture accordingly. So first, I would like to request Mr. Hare Krishna Swami, Scientific Staff, Botanical Survey of India. And all the virtual attendees are requested, please be online and don't waste time in connecting yourself. So stay online so that you can immediately switch after Mr. Swami.
Good afternoon to all of you, everyone. So the chairperson, my seniors, juniors, and friends. Myself, Hare Krishna Swami, and now I am working as a scientific staff in Botanical Survey of India. The work that I am going to present, that is not really related to my present position, because recently 3 to 4 months back I have joined there, and all the works are related to my PhD work. So my topic is chlamydospore formation in trichoderma and its utilization for the sustainable rice crops. Okay, in introduction, we got that the species of the genus Trichoderma is most useful in the biocontrol of in many of the diseases of rice and crops, and this has a huge human impact. And the Trichoderma species have major impact on agriculture, known to enhance the growth and control of soil-borne plant pathogens. In India, it is alone more than 250 formulations are available, but almost all of them are based on the single strain, that is the Trichoderma viride. And why we go for the chlamydospore? Actually, many of the research works are done on the Trichoderma conidiospores, but there is a little work or no work has been done on the chlamydospore. Chlamydospore is the resting structure which is useful in the adverse condition of the Trichoderma. And the conidia are reported to be unstable when released in the environment, and they are sensitive to the high temperature and UV radiation. So it is better to come out with a better alternative, that is the chlamydospore. Chlamydospores are produced in harsh conditions in various fungi and maybe in resting adverse conditions. Please next. Challenges. What are the challenges we face during the time of chlamydospore work? First is the main problem is the chlamydospore is most unstable. Most of the Trichoderma species doesn't form chlamydospores naturally. Number of workers have tried induced to chlamydospore, but we have a very little success. However, the knowledge associated with the mechanism of chlamydospore formation and its biocontrol activities as compared to the conidiospore remain unclear in Trichoderma. Next. With these objectives, we have proceeded. First, we have artificially induced the chlamydospore in Trichoderma. Comparative analysis of chlamydospore and conidiospore in their biocontrol activity. Studies the mechanism behind this in the formation of chlamydospore in Trichoderma species. Next. So, in our study, what I have taken, all the in India now, all the works related to Trichoderma are based isolated from the soil. But we are first in India to report all the Trichoderma which have used are isolated from the tree bark. Here we have taken 7 species of Trichoderma, that is Trichoderma hergianum, Trichoderma erinaceum, Atroviride, Havensis, Pararesi, Trichoderma longibactum, Trichoderma resi. And here I clearly mentioned there's this Trichoderma erinaceum and Trichoderma Havensis, Pararesi and Trichoderma resi as first time reported in the India. Next please. And these are the pictorial images of various Trichoderma strains. Next. And this is the morphological structures of Trichoderma hergianum. Next. This is the morphological various spore structure, conidiospore structure, chlamydospore structures of the Trichoderma erinaceum. Next. This is from Trichoderma atroviride. Next. This is from Trichoderma havensis. Next. This is from Trichoderma pararesi. Next. This is Trichoderma longibacterium. This is Trichoderma resi. Next. And these are the morphological studies we have gone through. After that, we gone through the molecular characterization by using ITS, internal transcript spacer regions, and TF, translational elongation factor region, and RPV, RNA polymerase binding region 2. After getting all these 3 primers classification, we gone for the phylogeny FR. Here you can clearly mention that all the Trichoderma pararesi and our isolate is also in their group, and it is clearly distinct that all the species are no confusion. They are completely separated from each other and very well classified. Next. Okay, now we go for the induction of chlamydospore species in the identified trichoderma species. For that, we have taken 6 media, that is the oatmeal media and cornmeal media, corn stiff liquor media, corn and soybean meal media, Gorodka media, and potato dextrose medium. Here, in case after studies with the 7 species, we found that potato dextrose, the chlamydospore formation is very less in case of potato dextrose media, and it is very high in case of cornmeal media. Okay, next. And the genes, for that, after permission of the chlamydospore, we gone for the genetic study. What are the genes responsible during the chlamydospore formation in that trichoderma species? Especially, we have studied the glutathione S transferase, oxidoreductase, thiamine biosynthase protein, and demethylase, glycosyl transferase, peroxidase, catalase, proteinase, and DDS, okay, and chitinase. In that cases, we see that this glutathione S transferase, oxidoreductase, and thiamine biosynthesis proteins, and that is our demethylase, and some of the genes are down regulated during the formations of chlamydospores in trichoderma species, and some genes are up regulated. So this is the main difference between that. This study is the first report in India, and this work is now under evaluations of the research paper. So I cannot details go through it. So this is the genes which are responsible, down-regulated and up-regulated during the formation of chlamydospore. Next, please. So what are the enzymes responsible for the formation of chlamydospore? We are mainly studying in the endoglucanase, cellulase, peroxidase, catalase, and superoxide dismutase. And in all the cases, we have seen that the trichoderma erinaceum, which that is CRRI T2, and trichoderma and trichoderma hergianum, they are mostly responsible for these enzymes, and these are the various enzymes responsible for the chlamydospore formation in trichoderma species. Next. And this is the study of a viability of chlamydospore-like structures. We have prepared the chlamydospore, we have induced the chlamydospore. Now, after that, we have gone for the formulation, how the farmers will use that chlamydospore. After formulation, we have after going for the formulation, we have tested how many days they have their lifespan. So for that we have studied, and we take that it is per till 180 days. As compared to the conidiospores, the chlamydospore formulations are very high self life, and it is very good for the farmers field. Next, please. Next. Next, please. No, it is not going on. No, it's not moving. Yes, yes, yes. I will wind up. Yes, 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 madam. Okay. After that, we are going for the mainly seed blight disease of rice. We have this is the control plant of seed blight disease, and here we have given the seed treatment of chlamydospore, and here we have given the chlamydospore seed treatment, and here we have given the chlamydospore of a foliar spray, and here as compared to the diseased plant, and all it's in that all these treated plants, the disease is very less. Next, please. And here also in trichoderma, CRRI T1 spore foliar spray and chlamydospore foliar spray. Next. And this is also CRRI T2 seed treatment of chlamydospore, and this is CRRI T2 spore treatment. Next, please. Next. No, scroll it. Okay. Now it is the disease progression curve, and this is in the disease progression curve. You see that in the trichoderma chlamydospore foliar spray, the disease progression curve is very less as compared to the others. Next. Okay, these are the various enzymes that are responsible during the process of disease and trichoderma chlamydospore interactions. This is mainly enzyme activities, catalase, peroxidase, and superoxide dismutase. Next. These are the various growth promotion parameters of trichoderma seed treatment, chlamydospore. Next. And this is also after 21 days of sowing. This is the enzyme activity in case of chlamydospore and the control ones. Next. And it is after just before the flowering. What is in the active tailoring stage? This is the enzyme activity. Next. And this is the various parameters are related to growth parameters or agronomical parameters. We have here we have completely seen that the trichoderma chlamydospore seed treatment and spore treatment are at par equal. Next. And significant findings what we have get there. For the first time in India, we have reporting Trichoderma erinaceum, Trichoderma havensis, Trichoderma pararaceae, Trichoderma raceae. And the entire investigation provides that the maximum diversity of this genus is occurring in the above ground. And the Trichoderma isolates strongest ability to produce chlamydospore, and no pigments, insoluble substances were observed in the oatmeal medium. And the potato dextrose medium may be adapted as the chlamydospore producing and chlamydospore non-producing media respectively. And Trichoderma havensis and Trichoderma erinaceum was superior antagonistic activity in major rice deep pathogens. Next. Another thing is that chlamydospore based trichoderma formulation is resistant to storage and control to the rhizoctonia solani also. The unigenes glutathione reductase, oxidoreductase, chitinase, demethylase are also responsible during the formation of chlamydospore. Unigenes peroxidase, catalase, protein kinase are also responsible during the formation of chlamydospore. CRRI-T2 seed treatment and CRRI-T2 chlamydospore seed treatment exhibited a higher activity as compared to other treatments. Conclusion: from all the above observations, it can be concluded that the chlamydospore is better candidate for the biocontrol of rice diseases, especially the soil-borne diseases. And it can be also concluded that the foliar spray gives better management for rice health as compared to the seed treatment. However, it is very necessary to compare the efficacy of chlamydospores and conidia under the field conditions. Okay, next. These are some selected references. Next. Oh, these are the some of my publications which are come from the Frontiers in Microbiology, Microbiological Research, and Environmental Science and Pollution Research. Next. And it is from again one paper from Environmental Science and Pollution Research and Journal of Environmental Management, Protoplasma. Next. And finally, my guide has also told that in the morning that we got the patent in the year of 2015 we filed it, and this year in the 2021 we have granted that patent. Next. So I am really thankful to Dr. Roshan K. Mukherjee Bark, one of the trichoderma specialists in India, for his timely cooperation, help and everything. Dr. Pradipta Mahapatra from Ravenshaw University, and Dr. Tota Nadak, scientist, ICAR NRRI, Dr. Kutubuddin Ali Mullah, ICAR NRRI, and Director, National Rice Research Institute for conducting my PhD degree, and Department of Botany, Ravenshaw University for providing me the registration facility here, and DST, Government of India, for providing me the Inspire Fellowship for 6 years for continuing my PhD degree. Now I am specially thankful to the Director, Botanical Survey of India, for allowing me to present my work with all the official entitlements, and also Professor Dr. Sudhanshu Sekhar Das, Deputy Director and Technical Section in charge, for his timely cooperation and everything and inspiration. Next. Thank you. Next. And this is my previous lab mates, which without whom I and my research work is completely incomplete. Okay, thank you the organizer for giving me the opportunity to present my work before this session. Thank you.
Thank you, Dr. Sai. Now I request the next speaker who will be joining online is Dr. Chitra Sood Kulkarni, the University of Horticulture Sciences. She may directly join without wasting time, and as it was evident in the morning session that she should represent it in the reduced mode so that the slides can be easily operable and without any technical delay. Dr. Kulkarni. Yeah. Welcome, ma'am.
Sure, madam.
I think you can run.
Start, sir. Good afternoon, delegates. Shall I start my presentation? Madam.
Audibility problem here. Sound is not properly being. Hello.
Hello. Hello, madam.
Can you start your presentation?
Yes, madam, I can start my presentation.
You can just address to, as Dr. Saab has told, you can just address what was your PhD problem and what was the work, and directly you can jump into the work done and conclusion.
Yes, madam, thank you.
Methodology part you can definitely skip because the result will speak about what you have done and how you have done.
Yes, madam, yes, madam.
Directly, without, so as to save the time. Okay, thank you so much.
Thank you, madam. Good afternoon, respected delegates. Good afternoon, respected delegates. I am Chaitra Kulkarni. I have completed my PhD in genetics and plant breeding from University of Horticultural Sciences, Bagalkot. My PhD research work was on transcriptome dynamics of Asiatic and Western carrots to decode the candidate genes for root adaptability. I worked under the guidance of Dr. Sarvamangala S. Cholin. Carrot is among the top 10 important vegetables, and it is considered as the second most important vegetable for its nutritional and economic wealth. Carrot is mainly consumed for its richest source of beta-carotene and carotenoid content and many other health benefiting factors like anti-cancerous, antioxidant and detoxifying properties and many other. And it is a model plant species for the somatic embryogenesis studies, and it is a first plant species approved for production of the biopharmaceuticals for various human health diseases. Globally, cultivated carrots are classified into two different types, Western types as well as the Asiatic types. All the historical records and scientific evidences prove that Western carrots are evolved from the Asiatic carrots. And over the period of domestication, Western types have developed into deeply pigmented root tissues, whereas Asiatic types are having the clear xylem phloem patterning over a period of domestication. As Asiatic carrots are adaptable to tropical region, they are also called as the tropical types, and Western types, they are adaptable to temperate region, they are also called as the temperate types. There are two important phases in the life cycle of the carrot. One is vegetative phase, in which secondary storage root development takes place. Another one is the reproductive phase. Using the stored energy in the secondary storage root, the plant will initiate flowering and seed production takes place. As the Asiatic carrots, they are adaptable to tropical climate. They are classified as the annuals. They don't require any vernalization period to switch from vegetative phase to reproductive phase. Whereas the temperate types, nothing but Western types, are classified as the biennials, as they require critical vernalization period to switch from vegetative phase to reproductive phase. Considering all these key important factors in the life cycle of the carrot, two important tissues, one is secondary storage root, which is the economic tissue, and another one is the seed governed by the floral primordia or the flower tissue. Both the secondary storage root and floral primordia were subjected for the transcriptome profiling in my PhD research work. These are the objectives of my work, in which a transcriptome profiling of the root and floral primordia along with its leaf between the temperate and tropical adapted cultivars by RNA sequencing, and differential gene expression analysis and identification of the candidate genes for adaptability as well as the flowering related genes, and the differentially expressed genes were also validated in the qPCR experiment. The two important genotypes representing tropical types and the temperate types were selected from the cluster analysis after the population structure assessment of the 144 Indian accessions, in which the two cultivars representing the tropical type and the temperate types were selected from two different clusters, and they were utilized for both vegetative phase experiment and the reproductive phase experiment. In the vegetative phase experiment, a secondary storage route was developed and it was subjected for sequencing, and in the reproductive phase experiment, floral primordia was produced in a phytotron facility with a temperature ranging from 5 to 7 degrees Celsius, and it was subjected for sequencing. And this is the plate showing the vegetative phase experiment in which the secondary storage route was developed. It was subjected for sequencing. Here you can clearly observe the clear xylem phloem patterning that is present in the Asiatic cultivar, whereas it is completely absent where the uniform pigmentation is present in the Western cultivar. To identify the differentially expressed genes responsible for this xylem phloem patterning, the vascular cambium responsive DEGs were identified after the transcriptome data analysis in my present research work. This is the experimental setup in which both the tissues were taken and it was subjected for sequencing. Total of 12 RNA-seq libraries were developed. Each 6 independent libraries were obtained from individual tissues like secondary storage route as well as the FPL tissue. And the transcriptome was further analyzed by different bioinformatic tools. A reference-based assembly as well as the de novo assembly was also constructed. A reference-based assembly was constructed using the high genome data, what is already present in the carrot, but the Asiatic cultivar, what we have utilized in our research work, has not included for developing the whole genome sequence data. Hence, a de novo assembly was also constructed to identify the novel candidate genes responsible for the root development as well as the flowering regulation. Then the differentially expressed genes were identified by different tools like a cufflinks suite for the reference-based assembly and the DSEC and HR tools for the de novo assembly, and the functional annotation of the differentially expressed genes was also carried out by David software in reference-based assembly and the Plastigo software, web-based software in the de novo assembly. And the gene ontology terms were defined for all the differentially expressed genes, and the gene regulatory network analysis was also carried out to find the GRN network for the vascular cambium as well as the flowering regulation. And but in present research presentation, I'm talking only regarding the candidate genes responsible for storage root development and vascular cambium patterning in the between the temperate and tropical cultivars. And these are the results. A total of 140 million reads were obtained from the root tissue. And the reference-based assembly confirmed the mapping rate of 73.87, and the de novo assembly also generated huge data in which genes and the transcripts were also obtained, and the Bosco assessment confirmed the highest completeness of around 89%, and the 98% of the completeness was also identified by mapping the individual libraries back to the generated assembly. And such generated assembly of the reference-based assembly and de novo assembly were utilized for identification of the differentially expressed genes. In the reference-based assembly, a cufflink suite could identify 3,544 differentially expressed genes, and in the de novo assembly, both the DSEC and HR could identify different number of genes. In order to avoid the discriminancy between the different tools, common DEGs identified between the tools were utilized, in which 2,239 DEGs were utilized for the downstream analysis. Here, this is the volcano plot representing the upregulated DEGs between the Western and Asiatic cultivars, in which the red dots indicates the upregulated DEGs that were identified for the Asiatic cultivars, whereas the blue dots indicates the upregulated DEGs that were identified for the Western cultivars. Different gene ontology terms were also defined for the various DEGs identified in the present research work. All the biological gene ontology terms defined for the biological process and KEGG pathway were in accordance, in which the upregulated gene ontology terms for the Western pathway were like photosynthesis, carbon metabolism, biosynthesis of secondary metabolites, plant hormone signal transduction, and various other domesticated genes what were present in the Western cultivars have been defined by the different gene ontology terms as well here, whereas the gene ontology terms were enriched for the Asiatic cultivars were like metabolic process and plant pathogen interaction, MAPK signaling, ABC transporters, and various others. This clearly indicates that Western cultivars are having the gene ontology terms is highly enriched for the photosynthesis, carbon metabolism, and secondary metabolite synthesis, whereas the Asiatic cultivars have having the geo terms specific for the MAPK signaling, plant pathogen interaction, and many other stress responsive genes. The same is also revealed with the biological process, and all the cellular component and the molecular function and related terms were also in accordance with these terms. And a different number of transcription factors were also identified for the root transcriptome data what we have generated in our work. A total of 477 transcription factor with the 45 different classes were identified by the root transcriptome data, and a total of 61 different transcription factors were also identified for the common DEGs what we have extracted between the Asiatic and Western cultivars. When we clearly see the types of the transcription factors what have enriched between the Asiatic and Western cultivars, we could see that Asiatic cultivars were having the transcription factor types like BHLH, BZIP, MIB, Ricky, NAC, and Matchbox, and other related transcription factors responsible for different stress responses as well as the flowering, whereas the Western cultivars were having the transcription factors like CO like AP2, GATA, GRF, GRAS, DOF, C2H2, and many other transcription factors involved in root growth development as well as flowering. C2H2 is the transcription factor which is responsible for FLC locus, which represses the flowering that was found abundantly in the Western cultivars, as Western cultivars required a particular vernalization period to suppress this FLC locus to initiate the flowering. This C2H2 was not present in the Asiatic cultivar. And a gene regulatory network was also constructed for the vascular cambium responsive genes between the temperate and the tropical cultivars to find the vascular cambium patterning enriched between these two cultivars. We could see that the networking what we have created could show that majority of the genes were interconnected with each other, which are involved in photosynthesis in the Western cultivars, whereas the root vascular cambium development related genes like TRN2, SPLN, HB6 were found to inhibit the xylem differentiation in the temperate cultivar. These inhibiting xylem differentiation types of the vascular cambium responsive genes were not present in the GRN what we have constructed for the tropical cultivar. In the tropical cultivar, the GRN network could show that majority of the genes were interconnected with each other, which are particularly involved in different stress responses like Ricky transcription factors, MIB, KIN3, NAC, and NLA, and many other transcription factors known for various stress responses were present in the gene regulatory network constructed for the Asiatic cultivar. And in the Asiatic cultivar, the genes like PERK and HCA2 are known to form the xylem differentiation. That was what we observed, known to be a major regulator which is responsible for the xylem patterning in the Asiatic cultivar, that we could see in the GRN of Asiatic cultivar, which was absent in the Western cultivar network. And to validate our RNA-seq data, we could select the set of different genes responsible for different abiotic stresses like mechanosensis, polyubiquitin, and ubiquitin, and many other Ricky gene and many other genes were selected, along with some growth responsive genes like cytosine protein inhibitase and glycosyl transferase is known for the carbon metabolism, which were selected and to validate the data. And both RNA-seq data and qPCR expression was concordant with each other, and thereby validating the transcriptome data what we have generated in our work. And a set of different genes were also sorted, which are responsible for biotic and abiotic stress responses in the work, and in which we could see that a total of 41 different genes responsive against different pathogens were identified, which are enriched in the Asiatic cultivar. Number of genes were found to be abundantly expressing in the Asiatic cultivar, whereas in the Western cultivars, the differentially expressed genes responsive against biotic stresses were very least. Only nearly 8 were present, that too against only bacteria, fungus, and other. And Asiatic cultivars were also found to be abundantly expressing against various abiotic stress responses. Around 72 genes were acting against different 21 class of abiotic stresses like cold acclimatization, response to oxidative stress, hydrogen peroxidate, and water depletion, and many other responses. The Asiatic cultivars were having highest number of genes against abiotic stresses, whereas the Western cultivars were having almost half of the genes acting upon the least number of the abiotic stress responses. And there were the root developmental genes were also identified, in which the xylem development, xylem phloem pattern related formation of the genes were identified for the Asiatic cultivar. The xylem development and xylem phloem pattern determining genes were absent in the Western cultivars. And these are the genes identified for the flower regulation in the Asiatic cultivars. Asiatic cultivars are known to immediately switch from a vegetative phase to reproductive phase. They don't require any vernalization period. We could identify some of the genes responsible for vegetative phase change and regulation of the timing of the transition between the vegetative to reproductive phase, and floral meristem determinacy related genes, and all the important genes responsible for flower development as well as the seed development were abundantly present in the Asiatic cultivars, whereas in the Western cultivars, the responsive genes for the flower development and the seed development were found to be least.
Dr. Chitra, could you please fasten and
Yes, ma'am, I'm
yeah, please. There is no sound. We can't hear you.
Madam? Madam,
Yeah.
you are hearing me?
Now we can hear, but your slide is not moving, I suppose.
Okay, madam. I'm almost complete.
Yeah. This is okay. We have already seen it. This is okay.
This is okay, ma'am.
This is also done.
Okay, madam. We could also identify some of the SNP markers in the root transcriptome data, which can be utilized for the QTL mapping and population structure assessment and marker assisted breeding and the genomic assisted breeding in the carrot. And with this present research work, we are reporting first time for the comparative transcriptome between the Asiatic cultivar and the Western types between the two economic tissues, that too particularly root and the FPL tissue transcriptome. Then the gene regulatory network, what we are reporting in our work is the first of its kind globally, and that can be utilized to reduce the undesirable core what is present in the Asiatic cultivars. And the huge RNA-seq data what we have obtained in the present research work can be utilized for improving the domestication related traits and root adaptability related traits by using the biotic and abiotic stress responsive genes what we have identified. We can use them for in the genomic assisted breeding programs, and the flowering pathway key genes that I have not reported in this present research, whatever presentation I have given, what we have identified, that can also be immediate utility in the crop breeding, and the candidate genes can be utilized for the genomic assisted breeding. And we could also identify some of the candidate genes and the gene products related to various human health diseases, like checkpoint for the cancer, like the PDL1 and PDL2 were identified, and the retinol dehydrogenase that is known for the eyesight improvement, that also we could identify, and the biotherapeutics can be done by using these genes, and many other genes also we have identified. These can be utilized in the molecular forming in the coming days. And with this, I would like to acknowledge the funding agencies like VGST that has funded for my present research work, and the University of Horticultural Sciences Bagalkot PG research grants, and also the lab facilities what I have obtained from the KBITS Department of ITBT Government of Karnataka, and for providing the doctoral fellowship, DST, PhD fellowship obtained from the KST EPS throughout my PhD research work. I would like to thank all the funding agencies and my guide for helping me to carry out this research work, and would like to thank the Agri Vision for helping me to present this research work in this platform. Thank you so much.
Thank you, Dr. Chitra. Now is the next presentation by Dr. D. Sai Charan.
Yes, madam.
Will he be presenting online, or yeah, it is shown online presentation?
Yes, madam.
Without wasting much of the time, directly you can switch over, and as suggested by Dr. Das,
Yes,
you can directly speak about your research problem and then results, and later on reaching to the conclusion. So don't waste time on explaining the methodology part. Directly reach the results, investigation done, and the conclusion. So that will be benefited by that one. Thank you so much.
Thank you, madam.
A very good afternoon to everyone. Can I start my presentation?
You can start your presentation.
A very good afternoon to everyone, respected delegates. Myself, Dharavas Sai Charan. I am a PhD scholar in Professor Jayashankar, Telangana State Agriculture University. And I am very thankful for Ravanshah University for giving me the opportunity to present my thesis. One of the objectives of my thesis entitled with the title, Potential Natural Enemies Associated with Sorghum Shoot Bug, Peregrinus maydis, ashmead. In this, I have identified about 20 natural enemies. Before slipping to that, we'll go into about introduction of our crop. Sorghum is a major important cereal crop which stands fifth next to all the cereal crops. And sorghum is one of the important crop in the northern Karnataka region. Unlike brown plant hopper, which is a major pest of rice, the sorghum shoot bug, the natural enemy associated with sorghum shoot bug under field conditions and their role in suppressing shoot bug population was not fully understood, although they belong to the same family. Further, the reports relating to natural enemies were scanty and need to be focused. In materials and methodology, during Rabi 2021, a Hattikunda susceptible variety of sorghum was sown in 20-20 meter square area in 3 locations at RARS Bijapur to attract the maximum number of parasitoids and predators towards host insect. The parasitoids and predators noticed on crop and also which associated with the Peregrinus maydis at different intervals were recorded, collected, preserved, and tested for predation under laboratory. And directly I'll flip into our results, where I have reported about 15 natural enemies, out of which the Anagarus species, Creontoides species, Tithias species, Erythraeus species, which is a mite, predatory mite, and Calitrachea species of a spider are most potential natural enemies associated with the sorghum shoot bug. Among them, Chilonimus exmaculated, which is a coleopteran, which is coccinellid. It able to feed on the nymphs, about 1 nymph per minute, whereas Creontoides species, which is a predatory bug, belongs to Hemiptera of Miridae, feeds on both eggs and nymphs, about 10 eggs and 1 nymph. Tithias species, which is also a predatory bug, reported to be feeding about 5 eggs per 1 nymph and 1 nymph. And Chrysopella carnea feeds about 1 nymph. And Erythraeus species is a predatory mite, which will be showing in the images, which stings adhering to the thoracic region of the shoot bug, but it is not found to be causing any lethality in the sorghum shoot bug. Whereas among the spiders which I have observed, Calitrachea species, Chicranthum approximatum, Marengo species, Neoscona species, Plexippus petersi, among all the spiders identified, all were found to be predatory, but among them, Calitrachea species, which is minute one and also abundant in nature, and it caused more damage to the sorghum shoot bug. And Anagarus species, which is a well-known mimariid smallest insect, which is found to be an egg parasitoid, found to be predator about 1 egg. And here we can see this is a mounted image of an Anagarus species, and this is Calitrachea species spider. And here we can see a reddish colored predatory mite, Erythraeus species, clinging to the thoracic region of the sorghum shoot bug. Thank you. I would like to thank my guide, Dr. S.S. Karamantanal, sir, who is the professor and scheme head to the AICRP on sorghum. And I would like to thank College of Agriculture, Vijayapur, for providing me lab facilities. And lastly, I would like to thank Ravanshah University.
Thank you so much for the brief and very crisp presentation of yours. Now I invite the next speaker, Dr. Mohanty. She is not available. I'm told that she is not available for her presentation. So I think we can jump over to the next presentation by Ms. Shruti. And she'll be speaking on profiling of the candidates of microRNA responsive to fluoride toxicity in rice. I think you can directly start your presentation. You can skip the methodology part. You can directly switch on to the title and the problem of your PhD work, and then coming to the discussion part directly.
Good afternoon, everybody present here. My name is Tamarpalli Shravya Shruti, and I'm currently pursuing my PhD in biotechnology from Ramadevi Women's University. So today I will be presenting on the topic profiling of candidate microRNAs responsive to fluoride toxicity in rice. Next. Okay. So rice is perhaps the most ancient crop ever known and the most vital crop that has ever been discovered. Rice, due to its ability to adapt itself under multifarious environmental conditions, has been contemplated as a strategic food crop by the FAO. The classical mutational and molecular breeding techniques have helped in development and increasing the production of rice through the development of novel varieties. But the production of rice over the last couple of decades has hit a plateau. Erratic rainfall, random climate changes, natural disasters, global warming, metal toxicity in the soil, and deadly attacks from pests can be termed as few reasons which are acting as a stumbling block towards world food security. Fluoride, a toxic xenobiotic compound, is a very important metal is causing a very important metal toxicity. It is an accumulative poison that reduces the overall biomass in plants by triggering the oxidative stress. As we can see here, this is a healthy plant, and once the fluoride starts accumulating in the soil, either in the plant, either through soil or groundwater, we can see what damage it causes to. It causes chlorosis and necrosis, decreases the chlorophyll and the carotenoid contents, increases the proline levels, produces reactive oxygen species, decreases the root and shoot length as well. So this brings, today there is no perfect strategy developed in order to control the fluoride toxicity. This is because due to lack of proper research in genetic, molecular as well as biochemical systems that regulate or govern the fluoride accumulation in the soil. So of late, microRNAs based regulation is emerging as an important molecular strategy that is being used in as that is being used in to understand how the fluoride toxicity decreases by using microRNAs. Next. MicroRNAs are nothing but a class of small non-coding RNAs which act as important modulators of gene expression related to several stress responses. These are the methodology that I have used in. Next. Okay, so the picture here represents, the first picture is of the sensitive rice genotype IR64, and the second photograph here is the tolerant aromatic variety Gobind Bhog. So in my work, I have used two rice genotypes, the IR64 and the Gobind Bhog. As you can see here, this is a representative of the image after 7 days of treatment, but we have collected the data on 15th day as well. And there we can see the leaves here turn completely yellow. The root and shoot length also thins as the number of days increases. And this graph here represents the fluoride concentration, and the blue bar represents the IR64 and the orange bar represents Gobind Bhog. So it is evident here that Gobind Bhog accumulates less of fluoride as it is a tolerant variety, and IR64 accumulates more of fluoride as it is a sensitive variety. The highest accumulation was seen at 25 milligrams per liter. Differential expression of rice mRNAs towards fluoride toxicity. So we have taken in 14 mRNAs which have proven roles towards multiple abiotic stresses, and out of those 14, 8 have differentially expressed and the rest 5 have not expressed. So mRNA 156, 166 and 171 were significantly induced in the fluoride tolerant Gobind Bhog. mRNA 160, 390, 396 and 444 were prominently induced in fluoride sensitive IR64, and mRNA 393 exhibited a basal response. Next. A total of 23 targets were predicted for 14 conserved mRNAs, and it was seen that most of the targets were encoded by transcriptional factors which have multiple, which act as regulators for multiple abiotic stresses. So these are the targets that are there. Say for example, mRNA 156, 160, 166 and 171 encoding the transcription factors which encode it are SPB box gene family, auxin response factor 18, and mate efflux family protein, scarecrow transcription factor family protein. All these are responsible for abiotic stresses such as salt, drought, and heat. mRNA 393, which encodes F-box domain and LRR containing protein, encode for abiotic stresses such as salt, heat, and metal stresses. And mRNA 528, encoding F-box domain and LRR containing protein, regulates for the same for heavy metal stress and salt stress. Next. The target genes that we had predicted when the expression analysis was performed. So a reciprocal expression was seen, which coincided with the targets that we have presented, making the fact clear that these particular mRNAs, particularly mRNA 156, 166, and 171, play a predominant role in regulation of fluoride stress in rice. Next. Next. Okay, these are the targets which were predicted, these are the targets that were validated through 5 RLM rays. So for a better understanding, the dark blue line represents the open reading frame. The light blue line is the putative mRNA interaction site. The red color nucleotides in the mRNA targets represent the mismatches in correlation with mRNA. And the arrow here, vertical arrow, represents the cleavage position in the mRNA. Say, for example, if we take this mRNA 396, the cleavage position is, say, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11th is the position for mRNA 396. Next. So I would like to conclude by saying these are the conclusions of my study, which says expression profiles of mRNAs and their targets suggest that IR64 and Gobind-Bog have differential response to fluoride toxicity. The mRNA 156, 166, and 171 showed positive regulation and presumably are involved in tolerance response of rice against fluoride toxicity. The study can be used as a solid foundation for further investigation of the roles of mRNAs when the rice is grown under fluoride stress. Next. What are the future prospects of this study? Since this is only a preliminary study conducted by me in the period of 3 months maybe. So these are the future prospects of this study, which I would be working on further during my course of PhD, which is identification of candidate rice genes or mRNAs regulating fluoride tolerance in rice, establishing the regulatory roles of mRNAs as modulators of fluoride signaling and tolerance in rice, development of host-specific target genes and mRNAs as markers for selection of fluoride-tolerant rice genotypes. Target genes and small RNAs can be developed, which can form the basis for future molecular strategies, which include some examples like genome editing, genomic transformation, towards engineering fluoride tolerance in rice. I would like to thank my supervisors, Dr. Rajkumar Joshi, Head of Department of Biotechnology, Ramadevi Women's University, Professor Sashmita Mohanty, Chairperson, PG Council, Ramadevi Women's University, ICR NRRA for providing me the seats, and lastly, Ravencha University and Agri Vision for providing me an opportunity at a very early stage in my career to present this. Thank you.
Thank you, ma'am. Now, the next speaker, Ms. Sonali Jaiswal. She'll be presenting her talk on plant growth promoting activities of endophytes isolated from drought-resistant plant Calotropis procera from the Gorakhpur University. She'll be joining with us in a short while. Now over to Ms. Jaiswal.
Am I audible?
Hello. You can directly start. Kindly check that if your slides are moving ahead and forward, because to save, so as to save the time. Otherwise, in between we get stuck. Yeah. I think in the same mode you can start without expanding them.
Okay. Good noon, everyone. Myself, Sonali Jaiswal, pursuing PhD under the guidance of Professor Sharad Kumar Mishra, Biotechnology Department, DDU, Gorakhpur University. Today, I will be presenting on my research, one of the research objectives, that is plant growth promoting activities of endophytes isolated from drought-resistant plant Calotropis procera. So we all are familiar with the global population increase. Thus, we would need to thus we would need adequate food resources to sustain. And also, the drastic climatic changes affects the agricultural crops and threatening the food security. So, how should we enhance the food or crop production for increasing population? So there are ways such as increasing agricultural land, but again that is limited due to the limited resources. Second option is the greater use of chemicals, but it is against eco-friendly approach. And third is more farm mechanization, again that is a very expensive approach. And the use of transgenics causes the ethical concerns. And the lastly is the use of endophytic microbes. Yes, this can be a better eco-friendly approach, cheaper and without any ethical concerns. So what are endophytes? Endophyte was first given by the Anton de Berry in 1866. And endophytes are the microorganisms that inhabit the plant organs without causing any harm to the host plant. And almost every plant contained at least one plant-associated endophyte. So endophytes can be endosym, that is endosymbiont, or endosympath, that can exist as endosyms or endopath, or endopath, that is pathogen. So endophytic microbes can be viruses, fungi, archaea, bacteria, protists, and they can inhabit the root, shoot, or leaves of the plant. And they help in plant growth promotion by producing the phytohormones, and in biofertilization process, that is solubilization of phosphate, nitrogen fixation, and siderophore production. And it also helps in biological controls of the pathogens. So endophytes and endophytes also have their functions in the production of bioactive compound, and the bioactive compound have the antioxidant, anti-diabetic, anti-arthritis, anti-inflammatory, anti-tuberculosis, antiviral, immunosuppressive, and anti-malarial and anti-cancer activity. So my research work focus upon the plant that is Calotropis procera, which belongs to the family Apocynaceae, and it is a drought-resistant, salt-tolerant plant that grows in sandy soil in the area of low rainfall. It also has it has been detected the presence of USP genes, that gene that provide the drought resistance in the plant. And the plant's part, just like root, bark, leaves, flowers, and plant exhibit anti-malarial, hepaprotective, anti-inflammatory, antipyretic, analgesic, and antimicrobial larvicidal activities. So next is isolation of endophytic bacteria from root, stem, and leaf, and endophytic fungus from the root, stem, and leaf of the Calotropis procera. So a total 20 endophytes were isolated, that is 14 bacterial and 6 fungal from the root, stem, and leaves of the Calotropis procera. Here we can see in the figure that in the control there is no growth of the microorganism, and in the sample part where the part of the stem and root were cut, so that their internal tissues be exposed on the media for obtaining the endophytic microbes. Next is all the bacterial and fungal isolates were assessed for the plant growth promoting traits like IA production, ACCD aminase production, ammonia production, phosphate solubilization, and siderophore production. So in here we can observe that the maximum plant growth promoting traits were shown by the CPR5B strain. And in case of fungal, The CPL1F strain showed the IA production is CDMNase, ammonia, phosphate, and siderophore production. And the molecular characterization was done by sending the culture to MTCC. And for bacteria, 16 sRNA sequencing was done, and for fungus, ITS region sequencing was done. And the sequence were submitted to NCBI Gene Bank, and accession number was obtained. Further, the effect of endophyte inoculation on plant growth was assessed in wheat plant, and here we can see that with irrigate and the experiment was divided into two treatment, that is with irrigation and without irrigation. That is providing the one is without drought stress and second is with drought stress. And here we can see that in control, when we compare this control to CPL1 if this strain, that is Penicillium citrinum, and CPR5B strain, that is Enterobacter cloacae strain. And here we can see that in comparison to this, the root and shoot length was increased. And same in the case of fresh and dry weight, where in case of control, when in comparison with controls with CPL1F and CPR5B, the fresh weight was a little increased. Next, effect of endophyte inoculation on plant growth in the non-irrigated plant, that is providing the drought stress. Here we can, if we compare with the control plant, we can see that in the root and shoot length were increased in the case of CPL1F when it was inoculated with it, and CPR5B when it was inoculated with Enterobacter. In the both case, it was highly increased in comparison with CPR5B, that is when it was inoculated with Penicillium citrinum. And same in the non-irrigated, here we can see that with comparison to control, the fresh weight was increased when it was inoculated with CPL1F, that is Penicillium citrinum, and CPR5B. Future perspectives, identification of endophytic species involves. So here we can identify the endophytic species which are involved in the enhancing the plant growth and developing the effective microbial formulation for boosting the plant performance. Gene expression studies with respect to tolerance induced by endophytes, and exploring the role of endophytes for discovering novel bioactive compounds in pharmacology, and commercialization of endophytes as biofertilizer. Thank you, everyone. And I would like to thank my DDU Gorakhpur University for providing me the lab facilities. And I would also like to thank Revin Shaw, AgriVision, for giving me the opportunity to present my work here. Thank you.
Thank you, Ms. Jaiswal, for a very crisp presentation on the topic of your PhD. Now, again, the next presentation is also online by Dr. Abhay Tiwari. It will be on the lactobacillus mediated fermentation of UVB irradiation mushroom powder to augment nutracellular properties. Now you can directly start your presentation online.
Ma'am, I am unable to share my screen.
So what should be the best option? You just suggest us. Because we are just waiting for your presentation to hear from you.
Ma'am, actually, it's message is showing that host is has disabled me for sharing option.
So डॉक्टर साहब बताइए कैसे करवाएं इनको, या तो वर्बल करवा दें इनका, मतलब हम सुनेंगे.
Yeah, I can share. Yes, yeah, I can. Now it's there, ma'am. Yes, I can share. Yes, ma'am. Yes, ma'am. My slides are not moving. My slides are visible.
They are very much visible.
Yes,
now
okay.
We can start. Yeah.
Thank you to all the dignitaries present here. And I am the final year PhD student. I will be submitting my PhD thesis, and I'm working under Professor Satyavati Sharma in the Center for Rural Development in IIT Delhi. So I'll be presenting some of the objective of my PhD. project that was granted by the MFPI and the MFRP AIMS IIT. So my title of my presentation is Lactobacillus mediated fermentation of UVB radiation mushroom powder to augment the nutraceutical properties. It's a greener approach towards the green towards the demand of the nutraceutical in it's not moving, ma'am. So starting with the sustainable development goal, which given by the UNO, uh, and one of the sustainable development goal is the good health and well-being, and how that can be achieved I will be stating in my coming slides. So the mushroom cultivation is booming because since morning we are discussing how to increase the farmers in income and how to just double the income of the farmer. So in that perspective only, we do not forget the farmers who don't have land and who are ultimately they're depending on the some other business like mushroom production, fruit and vegetable like these. So how to help or strengthen these farmers to augment the nutraceutical property so that the product quality and terms of the economic terms, they get better revenue. So mushroom production has been done in the India since since very long Asian time, but nowadays it has been boom because of the government policies and all. So the major variety in the India's grows in the like Lentinus edotis, pleurotus, and the Volvera. So why I have taken, I'm just jumping into the literature review, why I have taken this topic. So why mushroom is very good for us to have the bioavailability of the nutrients, because it contains the ergosterol, which is a precursor molecule for the vitamin D, and it have several bioactive compound like polyphenols, lectins, flavonoids, and it's also have the prebiotics, which are very well known for the growth of the probiotics. So they contain alpha glucan, beta glucans, and they also have n numbers of the dietary fibers which stimulate the growth of the probiotics. So that's why the this aspect haven't been explored, that to utilizing the mushrooms as a prebiotic source for the growth of probiotics, so that the nutraceutical augmentations of the probiotics like short chain fatty acid, organic acids, and antimicrobial properties should be enhanced. And we know that the probiotics are rich, having a very good impact on the health. They are generally regarded as safe and they enhance the immunity. And coming on to and the technology which we have developed in the present study was, first is to biofortify the vitamin D in the mushrooms, and then utilizing that vitamin D enriched powder to proliferate the growth of probiotics, and then evaluation of that probiotic powder in the osteoporotic mice model. Because until unless the developed product is reaching the market or it is having beneficial effect, it won't be help to the society. So to tell you that osteoporosis is the second global health problem after the cardiovascular, after the menopause cycle. Women after the menopause cycle, after age of 40 or 45 above, when deficiency of the estrogen occur, the osteoporosis lead to takes place in them, and because of that the fractures and bone pain and numbers of the side effects are there. So I have taken one example in in all the throughout the world region, the causes of this vitamin D deficiency and the osteoporosis is at high. And Asia, it's around 71.4%. So coming on, these are the general pathway, which when we have the vitamin D enriched product, then it follows the same pathway, and it is bioabsorbed in the first in the liver, and then it is get converted in the kidney, and then it is absorbed by the different part of the body. I'm not going into the detail of the pathway. So the recommended dose for the young people is that 600 IU per day, and along with the calcium when it takes, then it only get absorbed in the body. So vitamin D deficiency has an important consequence that it has the calcium. If inadequacy is there, then the calcium absorption will be lower down. Parathyroid hormone will be there, then the mineral density of the bone will be decreased, and the risk of fracture will be there. So how to address this problem to naturally take the organic food, and how to augment the vitamin D and the other bioactive component in the edible foods. I have taken the mushroom. So how to enhance the post-harvest quality of the mushroom, because mushroom is a highly perishable commodity, and because of the polyphenol oxidase which cause the browning in the mushrooms, and because of greater than 90% of the moisture is present in the fresh mushrooms, so the storage is a very big problem. So here I am suggesting that the augmentation of the vitamin D can be there by the exposure of the UVB. That can be traditional way, and then the artificial way, by utilizing that you can augment the vitamin D. One, you can use the sunlight, but the different the problem with the using of the sunlight is that it is having a wide range of spectra. It is having UVB, A and C, and the reach to the mushrooms, the direct effect on the mushroom will be not there, and the oxidation of the other bioactive molecule will be taking place. So here in the methodology we have selected the artificial source of UVB. So what happened is, when the mushrooms are exposed to the UVB light, and then it is having three initial precursor molecule called ergosterol, provitamin D3 and provitamin D4, and it is getting converted into pre-vitamin D, and then it is thermally converted into the active forms of the vitamin D2. And then this is the chamber which we have developed utilizing the UVB tubes and having the narrow band of 285 to 315, and by using the different exposure R time, with the distance of 10 centimeter from the exposure source, we have just kept the mushroom sample, and we have tried to augment the vitamin D2. So I am coming on to the result that after seeing that when compared to the control part, and after the 2 hours of the irradiation, the vitamin D got enhanced, and it is successfully fortified, and beyond exposure up to 2.5 years, the degradation effect is there, because I have shown earlier in the figure that it is a thermal reaction, and because of the other photobiotic product, the content of vitamin D2 got decreased. So it's a time temperature combination, and the distance of the source of the light is also important. So I have taken some source that they have utilized the UV sources, and they have enriched the mushroom with the vitamin D, and in our case it is around 49.3 microgram per gram of the dry weight. So apart from vitamin D, it also contained, I said, the polyphenol and flavonoid content. So it got also enhanced total flavonoid, and the total phenolic content has been increased significantly. And to assess whether it is having a radical scavenging activity or not, so ABTS and the DPPH and the FRAP assay was done, and they also depicted the significant higher value when compared with the control one. And since I have told you, it is also having a prebiotic compound, so it's a beta glucan. So beta glucan also got triggered, and it got enhanced by from 41% to 47% in the UV irradiated samples. Now the other content is the ergothioneine. Ergothioneine is also a very essential amino acid, which is not present in the either in the animal or in the plants. So mushrooms are very good source of the ergothioneine. So the ergothioneine content, although it got decreases, but the amount is not significantly got reduced, so it can be further can be utilized in the diet. And these are the profiling of the different amino acid. Interestingly, we have not found the methionine, histidine, and the serine content because of the like UVB degradation, or the drying of the mushroom has been initially done. So because of that, these content has been negligible. And the other bioactive compound have been studied using GCMS profile. So these are the spectra of the GCMS, and to name some, like pentadenoic acid and the hexadecanoic acid, arabinitol, fumaric acid, and ergostrol component. I told you these are the stimulating compound for the vitamin D2 synthesis, and they are also rich in the anti-cancer, anti-tumor, and the flavoring agent. They are used in the food industry, so they are also get enhanced in the UV treated sample. Now to come up with the probiotic properties, I have taken lactobacillus acidophilus because it is known for to tolerate weather, because mushroom are rich in the polyphenol, so they can bear the polyphenol, and mushrooms are rich also in the beta glucan. So I have just used first lactobacillus, and their growth profile has been observed, and then the plate study initial have been whether these bacteria are able to survive in the powder or not. So these studies has been done, different concentration of the powder have been taken, and then growth has been checked. Now these are the plate study, and then the broth study, different powder concentration. Here you can see the different ratio, and then time are from the 0 hour to up to 96 hour or more than 96 hour. I have just evaluated the morphology, colony forming unit in the MRS media, and I have optimized that 2% from 2% to the like 12% is very suitable because of the nutrient availability also, and it get properly dissolved in that. So UVB irradiated mushroom powder shows significantly higher CFU count. So until unless it having proved that whether it is having some beneficial effect or not, so that has been evaluated in the in vivo study by the help of the AIMS. So first, I have developed the osteoporotic mice model just by removing the this ovary, so that of the mice, so that the estrogen deficiency can be take place, and the osteoporosis can be induced in that mice. So these are the in vivo trials, and then I have taken different groups, and up to 6 weeks I have demonstrated the probiotic powder and of the LA and the mushroom powder, and different groups were there. There was sham, that was a positive control, OVX, that is the negative control, and OVX plus LA, and then OVX plus mushroom plus non-irradiated, and the irradiated one is given. And the CFU count was initially was at 10 to power 8 CFU per element, and 20.21 mg per body weight per day dose was administered to the mice. So here you can see after giving the different doses in the sham and the OVX, first the bone histomorphic parameters was checked. You can see the osteoporotic bone has having very degrading effect on the bone, and then the control one is there. And then when I have compared with the macrosity of the different dose, that is sham, OVX, LA1, and the non-irradiated and the irradiated mushrooms, you can see the therapeutic effect of the dose which we have given to the trabecular bone and on the collateral bone is there. Then the bone mineral density is also one of the other parameter which suggests that whether it is having impact or not is also done. Then we can say that a normal bone can be healthy if we eat the probiotics along with the probiotics, and then you can treat the post osteoporotic thing. Now, until unless the product is not since we have used two technology, one is the to biofortify the vitamin D2 content in the mushroom powder, another one is the to augment the cell count of the probiotics and increase the nutraceutical value. So first, the farmer training has been done in the Dholana area of the Uttar Pradesh, and some initial training has been done on how you can augment the vitamin D content in that, and how you can use day-to-day use, you can use that powder to biofortify in the dahi, milk, chach, so that the adequate amount of powder can be given to the women self-help group or the person who are suffering from the osteoporosis. So in the conclusion, I have to just say that the present technology is a sustainable technology to improve the nutraceutical potential of the dried powder in the mushrooms, and which holds significant in the diet. Since in the COVID time, we also observed that in the pandemic time, vitamin D was the one of the key factors which was recommended by the doctors, and it boosts the immunity too. And other apart from vitamin D, other boosting properties are there in the mushrooms. And the adopted methodology have been recently also adopted or approved by the world agencies like EFSA and the FDA. So it's not a problem for the farmers to use that. And if you use the symbiotic combination of pre and pro, the health benefit will be there. So the future perspective will be there. Then I have to evaluate some other product. Like right now, I have just used some only drink, like fermented drink, and the traditional food product. The other product like bakery and the this dairy one, that can be further. I have to evaluate and I have to check the sensory aspect of all these, the future prospective food product which will be developed. So this is the graphical representation of all the study which I have done. Firstly, I have just enhanced the vitamin D2 content, and then the in vitro studies of the proliferation of the LA, and then the OVX mice has been taken for the osteoporosis evaluation, and the storage study of the feed by trial powder have been evaluated. So some part of the study has been published in the LWT, and the other established fact that mushrooms are also a very good source of the vitamin D has been published in the International Journal of Food Science. And these are some of the publications. And I wholeheartedly thanks my supervisor, Professor Satyavati Sharma from IT Delhi, Dr. Vasudha Sharma from Jamia Avdar, who have taken care of the food products, and Dr. Rupesh Srivastava from AIMS, who has literally helped me to just, because my research will not be able to reach up to that level when it has not have been valid for its nutraceutical and the health aspect of the powder. I want to thank lab members of the IIT, Jamia and the AIMS, and I have to special thanks to the Ministry of MHRD for my fellowship, MOFPI, and for the MFIRP for granting this project and providing the financial benefits. Thank you.
Thank you, Mr. Tiwari, for a very illustrative and knowledge enhancing presentation on mushrooms and how we can enhance the osteoporosis problem in the country. After your presentation, all 6 presentations which were made in the Young Investigation Forum post lunch are open for question and answer session. So any of the participants who have some questions can raise their hand, or they can put up the questions, and all the presenters can answer those questions directly. So I think if we do not have any questions for this particular session on Young Investigator Forum, we can, Dr. Sahab, conclude kar de? With permission of my chairman, we would like to conclude this session. And nicely, out of 7 presentation, 6 were presented and 1 presenter was not there. Out of these 6 presentations, diverse topics on different aspects were covered. And these presentations are directly or indirectly related to agriculture enhancement and in doubling the income of farmers, which was reiterated today morning by many speakers in the morning sessions. So with this one, I just conclude and close this session, and wish you all the best for your future endeavors, particularly our young researchers who are very, very enthusiastic in working out and going for nation's cause and bringing out new research and putting everything on the reality platform. Thank you so much.